Hello, Alida this side. Hope this video finds you in good health. Um, Today in this video we are going to read about biotechnological applications in medicine. Okay, in my previous video we read about biotechnological applications in agriculture. So we are going to start with biotechnological applications in medicine. Okay, so the recombinant DNA technological processes have made immense impact in the area of healthcare by enabling mass production of safe and more effective therapeutic drugs. Further, the recombinant therapeutics do not induce unwanted immunological responses, as is common in case of similar products isolated from non-human resources. At present, about 30 recombinant therapeutics have been approved for human use the world over. In India, 12 of these are presently being marketed. This information is very much important. Okay, this information is very important. Next, um, genetically engineered insulin. Okay, management of adult onset diabetes is possible by taking insulin at regular intervals. We know the diabetic patients take insulin because diabetes is a disease which is caused due to under secretion of insulin by the pancreas. So diabetic patients take insulin at regular intervals and thus stay in control. How would a diabetic patient do if enough human insulin was not available? If you discuss this, you would soon realize that one would have to isolate and use insulin from other animals. Would the insulin isolated from other animals be as effective as that secreted by the human body itself? Would it not elicit an immune response to the body, since it would be a foreign material, right? So the body will make antibiotics against it. Hmm? Now imagine if bacterium were available that could make human insulin. Suddenly, the whole process becomes so simple. You can easily grow a large quantity of bacteria and make as much insulin as you need. Think about whether insulin can be orally administered to diabetic people or not. Why? Okay, the answer to this question I'll tell you, but at the end. Insulin used for diabetes was earlier extracted from the pancreas of slaughtered cattle. Okay, uh, from the pancreas of slaughtered cattle and pigs. Insulin from an animal source, though, caused some patients to develop allergy or other types of reactions to foreign protein. Okay, some patients developed allergies against this foreign protein. Insulin consists of two short polypeptide chains, chain A and chain B. Okay, chain A and chain B that are linked together by disulfide bridges. Okay, by disulfide bridges. So next is, okay, where is the part? Chain A and chain B that are linked together by disulfide bridges. In animals, including humans, insulin is synthesized as a prohormone, like a proenzyme. The prohormone is also needs to be processed before it fully becomes a mature and functional hormone, which contains an extra stretch called a C peptide. The C peptide is not present in the mature insulin and is removed during maturation into insulin. The main challenge for production of insulin using RDNA techniques was getting insulin assembled into a mature form. In 1983, Eli Lilly, an American company, prepared two DNA sequences corresponding to A and B chains of human insulin and introduced them in plasmids of E. coli to produce insulin chains. Chain A and chain B were produced separately, extracted, and combine by creating disulfide bonds to form human insulin. So, what does this mean? Okay, so what happens in insulin used for diabetic patients were earlier extracted from the pancreas of slaughtered cattle, okay, uh, and pigs. So, insulin from the animal source caused allergic reactions to some of the patients. For this rDNA technology, recombinant DNA technology, was used. In proinsulin, that is the inactive form of insulin, that is before maturation into insulin, in proinsulin, there is one A chain, the blue colored A chain, and there is this B chain. No, sorry. Blue color is A chain, green color is B chain, and there is an extra stretch which is called the C peptide. This color, this is the C peptide. So there is an extra stretch. So this A chain and B chain are attached to each other with the help of disulfide bridges. With the help of disulfide bridges. So when proinsulin matures to insulin, um, what is produced? What do we get? Chain A or peptide A and peptide B. Okay, and in the mature insulin, the extra stretch, that is the C peptide, is not there. In the year 1983, Eli Lilly, an American company, prepared two DNA chains or sequences, DNA sequences, which were similar to chain A and chain B, that is peptide A and peptide B, and what they did, that is they these two peptides, that is peptide A and peptide B and B, they took DNA sequences which were almost similar to or corresponding to these peptide A and peptide B, and introduced them in the plasmid of E. coli. Introduced them in the plasmid of E. coli to produce insulin chains. Okay, chain A and chain B were then attached to each other with the help of disulfide bridges to form human insulin. That's how human insulin was provided by Escherichia coli. Gene therapy. If a person is born with a hereditary disease, can a corrective therapy be taken for such a disease? Gene therapy is an attempt to do this. Gene therapy is a collection of methods that allows correction of a gene defect that has been diagnosed in a child or an embryo. Here genes are inserted in a person's cells and tissues to treat a disease. Correction of a genetic defect involves delivery of a normal gene into the individual or embryo to take over the function of and compensate for the non-functional gene. Okay, so now there is a case study of a 4-year-old girl. Listen. The first clinical gene therapy was given in 1990 to a 4-year-old girl with adenosine deaminase deficiency. Okay, ADA deficiency, adenosine deaminase deficiency. This enzyme is crucial for the immune system to function. The disorder is caused due to deletion of the gene for adenosine deaminase, gene for ADA gets deleted, and this disorder takes place. In some children, ADA deficiency can be cured by bone marrow transplantation. In others, and it can be treated by enzyme replacement therapy, in which the functional ADA is given to the patient by injection, just like insulin is given. Okay. So functional ADA This is the enzyme replacement therapy in which the functional ADA is given via injection to the patient. But the problem with the both of these approaches, that is the enzyme replacement therapy or bone marrow transplantation, the backdrop of these were that uh, mm, okay, that they are not completely curative. As a first step towards gene therapy, lymphocytes from blood of the person are drawn in a culture outside the body. Okay, lymphocytes are taken out of the body of the patient and are grown in a culture outside the body. Hmm. uh, Functional ADA cDNA complementary DNA. Okay, ADA complementary DNA using a retroviral vector is then introduced into these lymphocytes, which are subsequently returned to the patients. Subsequently induced into these lymphocytes, which is subsequently returned to the patient. However, as these cells are not immortal, the patient requires periodic infusion of such genetically engineered lymphocytes. Hmm. However, if the gene isolate from the marrow cells, producing DNA is introduced into the cells at early embryonic stages, it could be a permanent cure. Okay, so in the year in 1994, year-old girl with adenosine deaminase deficiency uh, was given the first gene therapy. This information is very much important. Whom the first gene therapy was given in 1994, year-old girl with adenosine deaminase deficiency. So this enzyme is actually crucial for the immune system to function, and this disease is caused due to the deletion of the gene for adenosine deaminase. In some children, the treatment is bone marrow transplantation, or in some, it is gene replacement therapy. But the problem is that in both the cases, it is not completely curable. So, the first step towards gene therapy was taken. So, how was it done? The lymphocytes from the patient's body was taken out, and um, uh, they were uh, grown in a culture outside, in vitro. Okay, in a culture outside the body of the patient. A functional ADA, that is adenosine deaminase complementary DNA, was introduced into using a retroviral vector, was then introduced into these lymphocytes. And since these lymphocytes are now made recombinant, like they have the recombinant DNA, these lymphocytes were returned to the patient's body. But these lymphocytes are not immortal. Okay, lymphocytes are mortal. So obviously this needs to be done from time to time, right? Again, it's not completely curable. Okay, so the patient requires periodic infusion of such genetically engineered lymphocytes. But if the gene is isolated from the bone marrow cells producing ADA, if the gene isolated from the bone marrow cells producing ADA is introduced into the cells at early embryonic stage, then it could be curable. Okay, genes isolated from the marrow cells uh, could be um, introduced into the cells at early em- into the defective cells in the early embryonic stage, then it could be a permanent cure towards ADA deficiency. Okay, next we have molecular diagnosis. What is molecular diagnosis? uh Here you need just these parts. Recombinant DNA technology. See, um okay, molecular diagnosis, right? Molecular diagnosis You know that for effective treatment of disease, early diagnosis and understanding its pathophysiology is very important. Uh, you don't need all those things. Okay, you need it from here. Recombinant DNA technology, polymerase chain reaction PCR, and enzyme linked immunosorbent assay, ELISA, are some of the techniques that serve as purpose of early diagnosis. Up to this will be enough. However, at very low concentration of bacteria or virus, that is from here, at very low concentration of a bacteria or virus can be detected by amplification of the nucleic acids by PCR. Okay, PCR can now be routinely used to detect HIV in suspected AIDS patient. It is being used to detect mutations in genes in suspected cancer patients too. It is a powerful technique to identify many other genetic disorders. Next is the hybridization technique. A single stranded DNA or RNA tagged with a radioactive molecule probe is allowed to hybridize to its complementary DNA in a clone of cells, followed by detection using autoradiography. The clone having the mutated gene will hence not appear on the photographic film because the probe will not have complementarity with the mutated gene. ELISA is based on the principle of antigen antibody interaction. Infection by pathogen can be detected by the presence of antigens, proteins, glycoproteins, etc., or by detecting the antibodies synthesized against a pathogen. From here, learn three definitions, that is recombinant DNA technology. You have learned it in the previous chapter only. Learn what is PCR. Okay, learn what is ELISA, and learn hybridization technique. Okay, which one is hybridization technique? This one, a single stranded DNA or RNA from here to mutated gene. This part is the hybridization technique. Learn this. Okay, all right. Okay, now we don't need the transgenic animals. Okay, uh next, omit the transgenic animals part. Next is the ethical issues. From ethical issues also, we are going to study only few portions, because from there only the questions are coming year after year. Okay, the part is this one. Ignore the first two paragraphs. Therefore, the Indian government from here to the public services. This part we are going to study, and here also there is nothing to explain, only memorization. This chapter is entirely about memorization. Therefore, ignore the therefore one. Okay, the Indian government has set up organizations such as GEAC, Genetic Engineering Approval Committee. Very important. Remember this. Um, the full form: GEAC, Genetic Engineering Approval Committee, which will make decisions regarding the validity of genetic modification research and safety of introducing genetically modified organisms for public services. From here, then again, uh, you need to come to rice. Okay, yes, rice. There are estimated, from here, there are estimated to, you need to study up to India. Okay, there are estimated thousands of years in Asia's agriculture history. There are sorry, there are estimated 200,000 varieties of rice in India alone. The diversity of rice in India is one of the richest in the world. Basmati rice is distinct for its unique aroma and flavor, and 27 documented varieties of basmati are grown in India. This much in it. Again, from in the year in 1997, from the here 1997 to novelty. Okay, to novelty. In 1997, an American company got patent rights on basmati rice through the US Patent Trademark Office. This allowed the company to sell a new variety of basmati in the US and abroad. The new variety of basmati had actually been derived from Indian farmers' varieties. Indian basmati was crossed with semi-dwarf varieties and claimed as invention of a novelty. So try to just think of it. uh, Basmati was India's own rice. Okay, Basmati was India's own rice. It was created by Indian farmers, but an American company all of a sudden got patent rights on Basmati rice through the US Patent and Trademark Office. So this is how they actually stole our native variety of rice. Okay, the basmati, the revenue that you pay by purchasing basmati chawal or basmati rice actually goes to the American company, not to our India. So try to think of it. Okay. Biopiracy. Now come to biopiracy. Biopiracy is the term used to refer to the use of bioresource by multinational companies and other organizations without proper authorization from the countries and people concerned without compensatory payment. Okay, this much. Again, the next para. Most most of the industrialized nations are rich financially but poor in biodiversity and traditional knowledge. In contrast, the developing and underdeveloped world is rich in biodiversity and traditional knowledge related to bioresources. This much is okay. Again, the last para. The Indian Parliament has recently cleared the Second Amendment of Indian Patents Bill that takes such issues into consideration, including patent terms of emergency provisions, research and developmental initiatives. That's it for this chapter. All this biopiracy, what is biopiracy? What step has Indian Parliament taken? Okay, the thing about basmati rice, memorize all those things. These are the portions from which the questions have been coming, and I can assure you no other questions will be coming from any other part of this chapter. And the topic transgenic animal has been deducted, so you don't need to study it. Thank you. Bye. Take care.